Hi, everyone. I'm Colin Wu from the Department of Chemistry, and I would like to thank Sarah Hosh and Settle for organizing this Teaching and Learning Symposium. The topic I would like to focus on today is integrating research experiences, or what I call authentic research, into our classrooms, and also use that as a vehicle to engage with the community. So just a little bit of history about myself. I am not shy to admit that I am by no means an expert in science education. My PhD is in biochemistry, and I always tell my students that I know a lot about very little. So I spent about 20 years of my life studying DNA repair mechanisms. I was fortunate enough to work with two absolute titans in the field as a grad student and as a postdoc. So both Tim and Maria are excellent mentors with virtually unlimited resources, so I could do whatever I wanted in the lab. But then the culture in the sciences is almost like Jedi training, right? So as a young apprentice, we are paired up with a senior scientist, either another postdoc or a graduate student in the lab, and we learn from them the day-to-day things. The PIs are integral in guiding the science, but they're not there for the day-to-day training type of things. So as I became a senior graduate student, or when I became a senior postdoc, I was the one tasked with working and training the next generation of grad students or postdocs in the lab. And that's actually one part of the job that I absolutely loved. And so when I was on the job market, I wanted to find a type of career that would allow me to not only do the type of research I wanted, but actually be involved in the lab in working with young scientists and training them and preparing them for the next step. And so when I became a faculty member at OU, my goals are really to train the next generation of scientists, but more than that, to just improve student attitudes towards the STEM fields in general. And this is, students at all career levels, whether it be at middle school, high school, university, college level, and just to get them active and involved in and get them excited about science. And one way to do that is to just increase their access to hands-on research, right? Allowing them to try things out in the lab to really see if that's something they want to pursue or not in the future. And then lastly, especially in this current age and climate, this is going to be an ongoing battle, and that is to build trust with the public and to bridge the communication gap between scientists and non-scientists, right? How do we communicate scientific information effectively at work and at home? And so I'm going to give a quick introduction about what I call, the dangers of cookie cutter experiments. So when I inherited the biochemistry laboratory curriculum, this is over exaggeration, but the experiments kind of look like this, right? So I equate lab work with cooking quite a bit. And that analogy works for the most part, but it does break down at some level, right? So in a way, when you look at a laboratory experiment or a protocol, it looks very similar to the recipe for chocolate chip cookies. You have a list of materials and then a list of procedures of how you assemble those materials together. And then presto, your cookies come out magically, right? And so at an intro level lab, that may be okay if your goal is to showcase how to make chocolate chip cookies, or in this case, how you purify a protein. So notice that is a lot more methods driven, right? Here you're trying to educate students on the technique, not necessarily about the fundamental science or inquiry that is behind those experiments. So on the upper level labs, I find that this is not really reflective of the scientific research process. And so students who do these types of experiments are going to be misguided about how research actually works. So one way to address this is to try to integrate research with education. So this process started around last year, and I was fortunate enough to receive some funding from the National Science Foundation to lead this project. And so the goal of the research, and pardon the jargon here, is really to try to understand how repair proteins that I study in my lab recognize damaged DNA structures. So that's the 30,000-foot view. And so we want to understand how repair proteins recognize and repair DNA in human cells. And we want to link the defects in DNA repair on their impact on heart contractile function. So that's the major focus of my research lab. But what I want to do is actually tie in these experiments into the biochemistry lab curriculum. And part of the motivation for doing that, as I alluded, is so that we can actually put in real research in the laboratory where the results are not already known and the experiments are not scripted, right? I don't know beforehand how the experiments are going to turn out. That's part of the fun and excitement in doing that. Another motivation for this is that in the Department of Chemistry, we house around 200 chemistry and biochemistry majors. We have about a dozen research active faculty. So if you do the math, we can't really place students in labs if every student wants to participate research with a faculty member. But if we actually put in our research into our classroom, that is a way to actually introduce a lot more students to research and get them excited about it. So in my biochemistry lab, what we do is there are literally dozens or hundreds of mutations in these DNA repair proteins that have unknown effects. Some of these mutations could cause cancer. Some of these could be completely harmless. And so what I have students do is actually use computational tools where they can predict and view what the structures of these proteins look like with and without the mutation and align them, right? So here we see a structural difference with and without the mutation. So the students are tasked with predicting what that mutation might do in terms of its DNA binding or its DNA repair activities. So they're formulating a hypothesis. It may be correct, it may be incorrect. That's okay. The goal is to then go into the lab and actually test those predictions. So they're going to engineer and purify their favorite mutant protein that they've been assigned. And then they are going to measure those protein DNA interactions themselves, and then write up their report and findings in the format of a research manuscript. So we've only done this for a few semesters, but I think it's been well received so far, where students get a lot more out of it. And even when the experiments don't work and we're in troubleshooting mode, students end up learning more from that process rather than just blindly following a list of procedures and have the experiment just magically work every single time. Okay. And then another component of this is public outreach. And so the goal of this part is to provide local area high school students and teachers with convenient access to research equipment, right? So high school teachers or high school classrooms, they don't have all of the instrumentation available in our research labs. So one way to address this is actually go and visit them with a portable instrument. And so I found this a few years ago. It's called a bento lab. It looks like has a footprint of a beefy laptop, but it combines a gel electrophoresis system, a PCR thermocycler, and a centrifuge all within that footprint that I can put in this, half height milk crate type thing. And so we can stack up 4 of these, put it in a cart, and actually take this to high schools so that students can now be able to do genetic testing. So we started this program last summer, where we visited Adams High School and Stony Creek High School. And what the students do is they swap their own cheek cells and isolate their own DNA. So these experiments are more fun when it is your own samples, right? And so in the first day, they isolate their own DNA and set up a PCR reaction, just like you would do to test for COVID. But in this case, they are testing for genes that are produced for processing lactose. So it's a genetics test for lactose tolerance. and we can do it right in a high school classroom with these portable instruments. So day one, they set up the PCR reaction. Day two, we come back and they run out their PCR products on a gel, and they can visualize it in real time and talk about those results. And so thanks to the support from not only the NSF, but our research office at OU, we now have 8 of these instruments, so 2 of these setups, where we can potentially visit multiple schools at the same time or multiple classrooms within the same school on the same day. So we're limited not by instrumentation, but rather by manpower or volunteer power, I should say. And so I'd like to give special thanks to Sarah and to Gerard for helping me lead with some of these demos. And this is something that we're able to involve our students in the secondary teaching education program to give them exposure and introduction to working in a high school classroom and network with local science teachers. So in fact, we're going to be visiting these 2 schools again, but also expanding our efforts to Rochester High School and Pontiac High School this year. In doing the outreach activities, I found out about this funding opportunity also from the NSF called the RET, which stands for Research Experiences for Teachers. This is an established program for engineering. For biological sciences, it's relatively new. So this is still at the proposal stage. We're resubmitting this proposal this summer. But this program, the goal of this program is focused on the teacher. So it'll provide high school science teachers with a way to do research at OU. And so we're going to mentor them in research so that they gain authentic research experiences. And we talked about the importance of that. And then these high school teachers will also work with OU faculty who are experts in education to design and also implement a curricular project that they will put into their classrooms the following school year. So it's meant to be a research-intensive boot camp. The participants get a stipend for being involved, and they also get some support for their classroom. So let's say they need some equipment and supplies to implement their curricular project, they have the ability to do so. Thank you again for being here and for being passionate about education. Like I said, I know very little about science education, so I'm definitely looking forward to hearing your feedback. So please email me if you have any questions or comments. And thank you.